Medical nanotechnology, revolutionizing diagnostics. Introduction. Welcome to today's lecture on medical nanotechnology. In recent years, nanotechnology has emerged as a promising field that holds immense potential in revolutionizing the field of diagnostics. By leveraging insights from physics, chemistry, and biology, scientists have been able to create nanosensors that can detect and diagnose diseases at an unprecedented level of precision. In this lecture, we will explore 12 different types of nanosensors that are making a significant impact in medical diagnostics. So, let's dive right in. Quantum dots are nanoscale semiconductor crystals that exhibit unique optical properties due to their size-dependent electron confinement. These properties make them excellent candidates for developing highly sensitive nanosensors. In the context of medical diagnostics, quantum dot-based nanosensors have been employed for the detection of various biomarkers. By functionalizing the surface of quantum dots with specific ligands, these nanosensors can selectively bind to target molecules, such as cancer biomarkers, enabling their detection and quantification. Moreover, the tunable properties of quantum dots allow for multiplex detection, where multiple biomarkers can be simultaneously monitored. This capability has the potential to revolutionize early disease diagnosis in a faster and more accurate manner. Plasmonic nanosensors utilize the unique properties of surface plasmon resonance, SPR, to detect and quantify analytes without the need for labels or reporter molecules. These nanosensors consist of metallic nanoparticles or nanoholes that interact with light, resulting in a characteristic shift in their optical properties. When target molecules bind to the surface of these nanosensors, the SPR wavelength changes, allowing for the detection of analytes. The label-free nature of plasmonic nanosensors eliminates the need for complex labeling procedures, making them ideal for rapid and cost-effective diagnostic applications. Furthermore, the high sensitivity of these nanosensors enables the detection of low concentrations of analytes, enhancing their utility for early disease detection. Carbon nanotubes, CNTs, are cylindrical structures composed of carbon atoms arranged in a hexagonal lattice. CNT-based biosensors have gained significant attention due to their exceptional electrical, mechanical, and optical properties. These properties enable CNTs to serve as transducers in biosensing devices. By functionalizing CNTs with specific biomolecules, such as antibodies or DNA probes, they can selectively bind to target analytes, triggering a measurable change in electrical conductivity or optical signals. This change can be precisely quantified, facilitating the detection and quantification of disease biomarkers. CNT-based biosensors offer great promise in the diagnosis of various diseases, including cancer, infectious diseases, and neurodegenerative disorders. Magnetic nanoparticle-based immunoassays utilize the unique properties of magnetic nanoparticles to facilitate the detection of disease biomarkers. These nanosensors are based on the principle of immunoassays, where specific antibodies are used to capture and detect analytes. Magnetic nanoparticles are functionalized with antibodies or other specific ligands, allowing them to bind to target molecules. By applying an external magnetic field, the magnetic nanoparticles can be separated from the sample, facilitating easy detection. The detection of the captured analytes can be achieved through various methods, including optical, electrical, or magnetic measurements. Magnetic nanoparticle-based immunoassays offer high sensitivity, rapid detection, and potential integration with existing clinical diagnostic platforms. Nanostructured electrochemical sensors utilize nanomaterials to improve the performance of traditional electrochemical sensors. These sensors rely on the measurement of electrical signals generated by redox reactions between analytes and electroactive species. Nanostructured materials, such as metal nanoparticles, graphene, or nanowires, are incorporated into the electrode surface, enhancing the sensing capabilities. The large surface area and unique electronic properties of these nanomaterials allow for improved analyte adsorption and enhanced electron transfer. This, in turn, translates into higher sensitivity and selectivity in disease biomarker detection. Nanostructured electrochemical sensors offer great potential for point-of-care diagnostics due to their portability, ease of use, and cost-effectiveness. Semiconducting nanowire sensors have emerged as powerful tools for the detection and diagnosis of various diseases. These sensors consist of nanowires made of materials such as silicon or zinc oxide, which offer several advantages in terms of sensitivity and size. The high aspect ratio of nanowires provides a large surface area for biomolecule immobilization, thereby increasing the efficiency of target detection. Additionally, the electrical properties of nanowires can be modified by the presence of target analytes, leading to changes in conductivity or field effect transistor characteristics. This enables the sensitive and label-free detection of disease biomarkers, paving the way for early disease diagnosis and personalized medicine. Piezoelectric nanosensors capitalize on the piezoelectric effect, where the application of mechanical stress leads to the generation of electrical signals. These nanosensors typically consist of piezoelectric materials, such as zinc oxide or lead zirconate titanate, integrated into a sensing platform. In the context of medical diagnostics, piezoelectric nanosensors are employed to detect and quantify physical parameters, such as force, pressure, or strain, exerted on the sensor surface. These nanosensors find applications in biomechanical measurements, such as monitoring cell adhesion, cell migration, or tissue mechanics. By providing highly sensitive and real-time measurements, piezoelectric nanosensors contribute to a deeper understanding of biological processes and pave the way for innovative diagnostic approaches. Optofluidic nanosensors combine the principles of optics and microfluidics to enable sensitive and specific detection of biological analytes. These nanosensors employ microfluidic systems integrated with optical components, such as photonic crystals or plasmonic structures. By controlling the flow of analytes through microfluidic channels, the target analytes can be selectively captured and analyzed using optical readout methods. The interaction between light and the captured analytes gives rise to measurable changes in optical signals, allowing for the detection and quantification of disease biomarkers. Optofluidic nanosensors offer high sensitivity, rapid response, and multiplexing capabilities, making them valuable tools for disease diagnosis and monitoring. Nanoplasmonic resonance sensing utilizes the interactions between light and metallic nanostructures, such as gold or silver nanoparticles, to detect and quantify analytes of interest. By precisely engineering the size, shape, and composition of nanoparticles, their optical properties can be tailored to resonate at specific wavelengths. When target analytes bind to the surface of these nanoparticles, the localized surface plasmon resonance is disturbed, resulting in shifts in the optical properties. These shifts can be detected through various spectroscopic techniques, enabling the label-free detection and quantification of disease biomarkers. Nanoplasmonic resonance sensing provides high sensitivity, real-time measurements, and has shown promise in various biological applications, including cancer diagnostics and drug discovery. Graphene, a single atomic layer of carbon, has garnered significant interest in the field of nanosensors due to its exceptional electronic properties. Graphene-based nanosensors offer excellent sensitivity, fast response, and low limits of detection. By functionalizing graphene with specific receptors, such as aptamers or antibodies, these nanosensors can selectively bind to target analytes and generate detectable signals. The interaction between graphene and the captured analytes leads to changes in electrical conductivity or electrochemical properties, enabling the detection and quantification of disease biomarkers. Graphene-based nanosensors hold promise for various diagnostic applications, including infectious diseases, cancer, and personalized medicine. Nanobiosensors play a crucial role in the early detection and monitoring of neurological disorders, such as Alzheimer's disease, Parkinson's disease, or traumatic brain injuries. These nanosensors combine nanomaterials, such as nanoparticles or nanowires, with biological molecules, such as antibodies or enzymes, to enable the specific detection of disease-related biomarkers. The binding of these biomarkers to the nanosensor surface triggers measurable changes in electrical, optical, or mechanical signals. This enables the precise detection and monitoring of disease progression or the effectiveness of therapeutic interventions. Nanobiosensors offer great potential in improving patient outcomes and facilitating personalized treatments for neurological disorders. In recent years, advanced nanosensors have shown tremendous promise in improving the detection and diagnosis of cancer at early stages. These nanosensors utilize various nanomaterials, such as gold nanoparticles, carbon nanotubes, or magnetic nanoparticles, to detect cancer-specific biomarkers. The functionalization of these nanomaterials with specific ligands enables them to selectively bind to biomarkers, triggering detectable changes in electrical, optical, or magnetic signals. These changes can be precisely quantified, aiding in the early detection and classification of cancer. Advanced nanosensors hold the potential to revolutionize cancer screening, allowing for earlier interventions and improved treatment outcomes. In conclusion, the field of medical nanotechnology is witnessing remarkable advancements in the development of nanosensors for diagnostics. From quantum dot-based nanosensors to advanced cancer detection tools, each of the 12 topics covered in this lecture highlights the multidimensional capabilities of nanosensors in disease detection and management. As we continue to unravel the mysteries of nanotechnology, these nanosensors have the potential to revolutionize medical diagnostics, providing faster, more accurate, and personalized approaches to healthcare. So, let us embrace these cutting-edge technologies and explore their incredible potential in improving human health.